flagella. All right. So let us start with the structure of cilia and flagella. And before we start, we should just do a quick comparative account of cilia and flagella. So cilia and flagella. What does cilia and flagella? I mean, how are they different? Some points of difference between cilia and flagella are cilia are smaller, flagella are longer. Okay, cilia are smaller, flagella are longer. Cilia are numerous. Their number is, I mean, they are throughout the body surface. The number of cilia is more than the cilia and then the number of flagella. So flagella is just one to few, whereas cilia is numerous. Also, the location, the cilia is all over, whereas the flagella is only at the anterior end. Now, coming to the kind of movements that they show, what kind of movement is shown by the cilia? Cilia shows what kind of movement? Cilia shows.
Ma'am, it shows an oar-like movement.
Yes, it shows a rowing movement. It shows a rowing movement. You understand rowing movement. While swimming, you must have seen this movement. What happens in swimming? You put one of your hand in the forward direction, all right, so to cut the water. So you put one of your hand forward. That is called a power stroke. So what do you do when you swim? So you put your one hand forward, and that is a power stroke. And then what do? You bring that hand back, and then in the same direction, you don't do the next movement. All right, so you bring your hand back, and the next movement is against the water. That is the recovery stroke. You understanding what I'm saying? So one is in the direction, and the other, you just don't go back the same path. You don't go back via the same path. You take another path while you are doing the recovery stroke. You understanding what I'm saying? So that is the same kind of movement in the flagella. Flagella shows rowing kind of a movement, a swimming kind of a movement, where they just bring they just move in the outward direction, and when they move in the outward direction, they cut through water. This kind of a movement is called rowing movement. Haven't you seen the movement in case of paramecium? See, paramecium has cilia all over. I'll show you the diagram of paramecium. So paramecium has cilia all over, and cilia shows a rowing kind of a movement. Cilia shows a rowing kind of a movement. So what happens in this movement? See, look at this. So you can see cilia is present all over. Cilia is present all over. All right. Cilia is present all over.
Yes, ma'am.
And then see what happens over here. Look at the movement. First is the effective stroke. First is the effective stroke, and the second is the recovery stroke. Wait, I'll explain you. Now see, this is cilia structure. Cilia has got a famous structure, and it is popularly called as, paramecium has got this structure, and it is popularly called as the slipper animalcule. It is called the slipper animalcule. Have you heard about this common name of the paramecium? It is called slipper animalcule because its structure is like a slipper. So it's called slipper animalcule. Now see, all over the body of the paramecium, you can see cilia is there. And this depression in the structure is the gullet. This depression is the gullet.
Ma'am?
Yes.
Ma'am, Aaron has left the video, the class because he has poor internet connectivity. He will be joining in soon in some time, but it will take him some time.
It's okay. So see what happens in this structure. Now so look at this. This is the structure of the paramecium. Paramecium is popularly called slipper animalcule. And one of the sides, you can see this kind of a structure, this depression coming. This is the gullet. Now what happens inside the gullet? See, everybody see clearly. You can see cilia facing in the inward direction. So what is the importance of this kind of a cilia? You know what happens is they are present in an inward direction. So this creates a whirlpool like movement. See, for example, you take sugar in a glass, and stir it. You take sugar and water in a glass and you stir it. So when you keep stirring, doesn't a cone kind of a structure, a whirlpool kind of a structure appear in some time in the glass? Don't you notice that? Anything which flows in a current in a closed structure and you keep stirring it, you will find a cone kind of a structure develops, and all the particles come at the starting point of that cone. You understanding? Such kind of a movement is created by cilia. So the cilia which are present on the inner side of this gullet, so they create this whirlpool kind of a movement. All the food particles, they just come over here and then they are taken in by the gullet. You understanding? This kind of a movement is the movement by cilia. Understanding? So by this ciliary movement, so cilia has dual role. First of all, it helps in movement also. In case of paramecium, it is helping in movement also. So cilia has dual role. It helps in movement also, and it helps in obtaining nutrition also. Whereas flagella only helps in movement. So that whirlpool thing that I'm telling you, so cilia beats, when the cilia beats in an inward direction, so a whirlpool kind of a structure is created over here with the water, and in that whirlpool are caught these food particles, and these food particles then move into the body of the paramecium via the gullet. Understood? There are two kinds of movements I told you shown by the cilia. I mean, two ways in which the cilia shows movement. First is the effective stroke. See, all the cilia are bending in this direction. You can see all are bending in this direction. And then there is this recovery stroke in the opposite direction. So the effective stroke and the recovery stroke, this is the power stroke. So you can see water's direction is this way, and look at the cilia bending this side in the power stroke and then this side in the recovery stroke. So the direction of locomotion is this side. So this helps in movement of the animal forward. All right. So this is in short an example how the cilia moves. So what kind of movement is shown by the cilia and what kind of movement is shown by the flagella? That is very important. Cilia shows rowing movement. This power stroke and the recovery stroke, that is called rowing kind of a movement, whereas flagella shows an undulating kind of movement. Undulating means, how just you take a string and you just give it a stroke, these waves kind of a structure are created. So this kind of a movement is shown by the flagella. Understood? You can write on the chat box and tell me you've understood or not. So this is just, the differences. So cilia and flagella differ in their size, in their number, in their location, in their kind of movement. And you also have different kinds of cilias and flagellas. So after the structure, I'm going to tell you the different kinds of cilias and flagella. There is one kind of cilia that shows movement, the other kind of cilia that doesn't show movement. Flagella always shows movement, but it has got two different names based on the structure it has. All right. Okay, so let us now come to the internal structure of cilia and flagella. Now, internally the cilia and flagella, they have the same structure. Internally, cilia and flagella, they have the same structure. And what is there internally? Let us just discuss the internal structure of cilia and flagella. So cilia and flagella are made up of two important things. See, this is the internal structure of cilia and flagella. I told you internally they are same. Internally they have the same structure. So the internal structure of cilia and flagella consists of two important things. The first is the protoplasmic sheath. And the second is the axoneme. All right. So you have two parts in the structure of cilia and flagella. You have the protoplasmic sheath and you have the axial and you have the axoneme. Axoneme are also called, the other name of axoneme is axial filament. It is axial filament. Now see, this axial filament has 9+2 arrangement. Now axial filaments are made up of microtubules. They are made up of microtubules. Microtubules are special kinds of proteins. They are made up of microtubules, and microtubules are present in a definite pattern. Microtubules are present in a definite pattern, and that pattern or arrangement is called 9+2 arrangement. You know, all the eukaryotic flagella have this 9+2 arrangement. So 9+2 arrangement means 2 are central microtubules and 9 are peripheral. So you can see 2 are central and 9 are peripheral. I'm going to explain you the structure. Now what happens? What is the structure of each microtubule? What is the structure of each microtubule that we have to do? Now each microtubule, let us just talk about the peripheral microtubules. So coming to the peripheral microtubules, each peripheral microtubule is a duplet. See, it has got A part and a B part. All right. So each microtubule is a duplet, and how the structure is, we are talking about the peripheral microtubules, and I told you they are doublets. So they are doublets. That means they are made up of two units. They are made up of two units, an A unit, which is on the inner side, and B unit, which is on the outer side. Okay. So you can just make this a little bigger. So A on the inner and B on the outer. And the wall, so this is like, we are talking about the peripheral microtubules. They are doublets. All right. So they are made up of two units, A and B. And their wall, the wall of A and B, the wall of A and B, the wall of A and B is made up of, you know, protofilaments. For example, just look at this structure. If this is the wall of, just imagine this kind of a view, you're seeing it from up. So this is the view of one microtubule, one peripheral microtubule. So if you just closely see on the walls, on their walls, you will have two sets of proteins. You have this alpha protein and beta protein like this. So you have alpha and beta. So these are like the units that the wall is made up of is called protofilaments. It's called protofilaments. So the A unit is made up of 13 protofilaments. So you can just make, 13 protofilaments inside. So all around the wall you have protofilaments, and B has 10 protofilaments. So 13 protofilaments are there in the wall of, not exactly I've counted and made, but yes, 13 is the number in A and 10 is the number in B. Understood? So, and one more important thing, A has got two arm-like structures, a shorter arm and a longer arm with a curved end. You can see this. So let us just once more revise till here so that we're not confused. So the internal structure of cilia and flagella consists of a protoplasmic sheath and an axoneme. Axoneme's alternative name is axial filament. Axial filaments are made up of microtubules. Their arrangement is 9+2. 9+2 means 9 microtubules are peripheral based on location and 2 are central. We are talking about the peripheral microtubules. They are doublets. That means they have two units, A and B. A is made up of 13 protofilaments in its wall and B has 10 protofilaments in its wall. Additionally, you can see that A has two structures like this, two arms like this. One is smaller facing inwards and one is longer which faces outwards and has a curved end. So clear till here, are the peripheral doublets clear to you? Then we will talk about the central part. Now just try to locate this structure. See over here, you can see A and B. So we've spoken A and B and how it is placed. Now see, this is the peripheral arrangement. So these are present in the periphery and their number is 9. So 1, 2, 3, 4, 5, 6, 7, 8, 9. And how are they placed? A is on the inner side, B is on the outer side. So this is this one. And A has two arms, one facing inside and the other facing outside. One facing inside and the other facing outside. So can you see in the diagram? You can just see and make out. So this peripheral microtubule is clear to you. Now let us just speak about the central one. Now the central microtubule is made up of two subunits. They are also made up of two subunits. We're talking about this now. We are talking about these central microtubules. I'll take a different color so that you can easily differentiate between them. I'll take a blue color for this. So central microtubules, they are named as C1 and C2. All right. Each is made up of 13 protofilaments. Remember protofilaments on the wall. So C1 and C2 are made up of 13 protofilaments. Now, how do you differentiate it? C1 always will have 3 radiating structures coming out. I will just draw and tell you. And each is 250 angstroms in thickness. Each is 250 angstroms in thickness. So you can see they are like almost same, but how do you differentiate these central microtubules? So you have this C1 and C2. So C1 and C2 are the two microtubules in the center. Both have 13 protofilaments, but how do you differentiate between them? There is this 3 raised structures coming out from this C1. So the microtubule which has 3 radiating structures coming out is C1 while the other is C2. So here you can see C1 and C2. All right, C1 and C2. So is the structure of the 9 peripheral microtubules clear and the 2 central microtubules clear to you? Now, they have not shown the connection, but yes, there is this, there are cross bridges. In between. Okay. And the peripheral and the central microtubules are made up of dynein protein. So let us just locate what we can see in the diagram. So you can see 9 peripheral microtubules. You can see 9 peripheral microtubules. A and B. A faces inward, B faces outward with 2 arms. A has 2 arms, one faces inward, one faces outward. And center may you have this central microtubule. And the entire, the two central microtubule are connected by cross bridges. They are connected by cross bridges. All right. And they are surrounded by a structure. This structure is called the central sheath. You know, this structure is called the central sheath. So you can see the central sheath. So what structures are clear to you? Outer fiber doublet, outer arm is clear to you. Inner arm is this one, inner arm is this one. Looking a little fadey, but you know, and you have the central fiber is clear to you. Now outside what you have is the protoplasmic sheath. Outside what you have is the protoplasmic sheath. All right. So till here, did you understand? You just tell me, write on the chat box and tell me, did you understand till here or not?
Yes, ma'am.
Completely?
Yes, ma'am.
Okay. So we have done now about the peripheral tubules and about the central tubules. Now, how are they linked to each other? You can see in the diagram that there is linkage between them, and how are they linked to each other? So see, the A part, the A unit has got this inner arm, and the inner arm is connected to the B subunit. The inner arm is connected to the B subunit. So everywhere you will link them in this way. So the inner arm, this is the inner arm, and this is connected to the inner arm is connected to, this is the inner arm, and this is connected to the B subunit. You can see the inner arm is connected to the B subunit. The inner arm, this is the inner arm, this is connected to the B subunit. Inner is connected to B. All the time, inner is connected to B. This is inner connected to B. All right. So this is one connection. This is one connection between the subunits. And next connection is so these connections between the inner arm and the B is called, they are called linkers. They are called linkers, you know. So this connection is called a linker. Linker is between the inner arm and the B subunit. Inner arm and B subunit. Followed. So linker is A inner arm with B. Okay. So that is a linker, and the linker is made up of nexin protein. The linker is made up of nexin. This nexin that they are saying, that is the protein which is present in the linkers. Now, additionally, you also have arising from that A subunit, you have these radial spokes. You have these radial spokes. You can see that they have labeled over here, you know, radial spokes. And the these are the radial spokes. And they have these, you know, rounded structures which will strike the central sheath. All right. So arising from the A part, you have these radial spokes. You have the radial spokes. So this is the structure of the cilia and flagella. Both of them internally have the same structure. So let us just once more repeat. And we should also know the kind of protein. So while the A and the B and the central subunit, they are made up of dynein protein. They are made up of dynein protein. We'll write it somewhere. They are made up of dynein protein. The linker is made up of, the AB linkers. The AB linkers are made up of nexin protein. All right, nexin protein. So where is the linkage? Now AB linker is like, how are they linked? Through which subunit are they, through which arm are they linked? What is the kind of linkage, or what structures are linked between A and B? The inner arm of the A is linked to the B subunit. The inner arm of the A is linked to the B subunit. All right. So this entire structure forms a linked structure. So till here, have you understood? Just a second. Let me just write everything in detail once more. So what we have done? In short, if you know this is the plasma membrane and here is the flagella or the cilia coming out, so only a small portion of the flagella or cilia is in contact with the plasma membrane, and that is the protoplasmic sheath. So we've just drawn it outside. So a rough structure, if you want to draw, you want to see how are the linkers present and all these things. So you will draw two central microtubules with cross bridges, and one is C1, one is C2. How do you identify C1? It has got these three raised structures. And I've told you already they are made up of 13 protofilaments each. And then there is this central sheath surrounding them. This is this central sheath surrounding them. And then what do you have? You have the doublets. You have the peripheral doublets, and the peripheral doublets. So just make a you will just make 9 dots like this: 1, 2, 3, 4, 5, 6, 7, 8, and 9. So 1, 2, 3, 4, 5, 6, 7, 8, and you can make one over here. All right. So just rub this. So there are these these are these 9 peripheral microtubules. 1, 2, 3, and 4, 5, 6, 7, 8, and 9. All right. Now you have A which faces inside, B which faces outside. So all the time A faces inside, B faces outside. So you have A inside, B outside, A inside, B outside. A is inside, B is outside. So A inside, B outside. All right. So all the time you have this structure. And then what do you have? There are two arms. I told you, the inner arm and the outer arm. The inner arm and the outer arm. So you have the arms coming out from A. So one place I'm going to label it, this is A, this is B. And one place I'm going to show all the linkage. All right. So how are they linked? So where is the AB linker? So this is linked to this. So what I wanted to tell you is the inner arm of A is linked to B. So this is one linkage. And all the time you have radial spokes coming out from A. So all the time you are going to connect it with connect as in, there's a small gap. So there is this, so inner arm is linked to B. Again, inner arm is linked to B, inner arm is linked to B. So all the time you will link the inner arm to B. So inner arm to B, again, inner arm to B. So let us now label it. So you understood the linkages. So let us just once more label it and sort it all. So this is the AB linker. AB linker made up of which protein? Made up of nexin protein. All right. This is the radial spoke. This is the radial spoke. So this is the radial spoke, and this is the cross bridge. This is the cross bridge. This is the cross bridge. And I told you the central microtubules and the peripheral microtubules, they are made up of dynein protein. They are made up of dynein protein. So dynein protein is what is present in the central and the peripheral microtubules. All right. So all these connections are clear to you. And where exactly, all right, what is this? What is this? Just write the name of this structure and tell me on the chat box. The sheath surrounding these two central microtubules. Very good. Central sheath. Excellent. So that is central sheath. All right. So this is the structure of the internal structure of cilia and flagella. So how does it function? How does it function? So what happens is when these radial spokes, they strike the when the structure has to move, the radial spokes strike the central sheath, and when the central sheath is struck, the inner microtubules, they move, and because of this, the entire structure moves. All right, so like how a wheel moves, the internal structure, the one which is inside the central wheels first move, and then that causes the movement of the whole structure. So that is how this moves. And on this basis, you can also tell me that for a structure to move, this central structure should be present. So the kinds of cilia, now there are two kinds of cilia, stereocilia and kinocilia. Stereocilia does not move, whereas kinocilia moves. So what kind of an arrangement will be there in the stereocilia? Stereocilia is going to have, they are non-motile. They are present in the sensory cells like the gustatoreceptors, and I've told you in the macula and all those structures in the ear. So you will find that the stereocilia do not move. So they do not have this central structure. So their arrangement will be 9 plus 0. 9 plus 0. Did you understand? 9 plus 0. All right. So let us now differentiate between cilia and flagella on the basis of types, okay. So this structure, internal structure, is clear to you. What you get is how are the peripheral microtubules connected to each other, how are the central microtubules connected to each other, which sheath surrounds the central microtubules. Now where is the protoplasmic sheath? Protoplasmic sheath is just on the outer side, because there's only a small portion. There's only a small portion. Just a second. There is only a small portion of the sheath which is connected to it. So the protoplasmic sheath is on the outer side. So let us now quickly differentiate between cilia and flagella on the basis of structure, not internal. Internally they are same, but two kinds of cilias are their arrangement internally. Internally, both of them have 9 plus 2. Internally, both of them have 9 plus 2, but this is variable. But this changes based on the kind of cilia. Stereocilia, is non-motile. So it does not have that central part. So it is going to have 9 plus 0. But kinocilia is motile. So this is going to have 9 plus 2. So please be careful what they're asking you in the MCQs. Cilia does have 9 plus 2 arrangement, but if they are specifically asking you about stereocilia, then it will be 9 plus 2 arrangement. And stereocilia is present in the sensory cells, well as kinocilia is present in all those structures where movement is required, and cilia beats outward and causes movement of substances like the trachea, the fallopian tube, and all these structures. And flagella has 9 plus 2 arrangement everywhere, and there could be two types. Whiplash. Whiplash is when the structure is, hunter type, as in, the width is not same. You have more width on one side and less width on the other side. And the other is, which is very common, is the tinsel type. The tinsel type has got uniform width, but it has got these, bristle-like structures coming out. So this is the most common kind of flagella found in bacteria and all. Zoospores of algae has got both kinds of flagella. So zoospores of brown algae. You do get this question. Zoospores of brown algae have tinsel kind of flagella. All right. So this is all for cilia and flagella. So externally they may be different. The kind of movement brought about them would be different. But yes, internally the structure is same, and that is that wheel kind of a structure that they have 9+2 arrangement. But yes, be very careful when they ask you about stereocilia specifically has 9+0 because it does not show movement, so the central part is not present, and this is present in sensory structures. And this is present on the trachea, fallopian tube. Understanding? And this is present in zoospores or brown algae. I'm talking about tinsel and whiplash both. So are you clear with the structure of cilia and flagella now? Is there any confusion, any question related to this? Will you be able to do the MCQs or not? Just let me know if it's all clear or not.
Yes, ma'am, it's clear.
It's clear?
Yes.
All right. So then the last one, the last structure where you had the doubt is your centrioles, right? Okay. Now coming to the centrioles, let us just look at the PPT. What kind of a structure is present in the centriole? So this is the internal structure for cilia and flagella. So they give you just the internal structure. They will just give you this diagram and ask you about this. This diagram is there in NCERT also, right? So you can now see necks and bridges there. Outer fiber doublet is clear to you. Membrane, this is the protoplasmic sheath. Central fiber, radial linker is there. Central sheath is there. So now all the labelings are clear to you. Now coming to the centriole. So centriole and basal granule are two things whose internal structure is same. So generally they are taught together. So internal structure of cilia and flagella was clear to you. Now we're going to discuss the internal structure of centriole and basal granule. So before that, you should know what is a centriole, what is a basal granule. Now centriole, as you know, popularly it is present in the animal cells. And what is the position of the centriole? What is the position of the centriole? So if you have this, always we do the comparison between centriole and basal granule. Basal granules are the structures from which the cilia and flagella originate. So these are basal granules. Centriole, what is their position is perinuclear. They are around they are near the nucleus. So their position is perinuclear. Whereas basal granule, it is below the plasma membrane. So below the plasma membrane. So haven't you seen, if there is this plasma membrane is there, then you have these basal granules, and then flagella originate or cilia originate from the basal granules. So we're talking about those basal granules. They are two in number, and they have got two structures which are perpendicular to each other. So like, you will have this kind of structure. If this is one, then another centriole is going to be perpendicular to each other. And then you have these particles which are all around that I'm going to tell you about. And this is numerous. So what will be the number of basal granules? Obviously, the number of basal granules will be equal to the number of cilia or flagella. What is the diameter? What is the diameter of the centriole? Centriole is thinner. It's just 0.3 to 0.5 micrometers. Whereas this has basal granules have larger diameter. Basal body has larger diameter. So this is 300 to 500 micrometers, 300 to 500 micrometers. All right. So centrioles help in cell division. They help in, you've done cell division. What is the role of the centriole? They help in spindle formation, aster formation. Whereas basal granules, what do they do? Basal granules, what do? They help in movement of cilia and flagella. So they help in movement of cilia and flagella. So all the cells having cilia and flagella have these basal granules or basal body. So this is what they look, how do they look externally? Now, all the cells, all the animal cells have centrioles. Exceptional cases, what is the cell which does not have centriole? Being an animal cell, it does not have centriole. Come on, I want the answer from you. Can you tell me any animal cell which does not have centriole?
Nerve cell.
Yes, correct. And also mature RBCs. Mature RBCs, mature mammalian RBCs, they lose the centrioles. They lose their centrioles. Basically, they have just the cytoplasm and the membrane. They do not even have a nucleus.
It is done to increase the oxygen binding capacity.
Yes, I mean, no, basically they are like empty. They do not have any organelle. Nucleus is large, so it cannot be present in the mammalian RBC. Mitochondria is not there, otherwise it is going to start utilizing oxygen rather than transporting most of it. So mature mammalian RBC lacks the centrioles. So centrioles and basal body, this is the basic difference. Now internal structure is same. Internal structure is same, but you know what is there over here is internal structure is same, but what happens over here is they are present in triplets. They also have microtubules. So this is the internal structure. So in this diagram, you can see this is the centriole, and the area around the centriole is called the centrosphere. So this is the these are the centrioles. I told you, two perpendicular to each other, and they are made up of microtubules. And so this is one centriole, this is another centriole, perpendicular to each other. And each centriole is made up of three triplets. So I told you doublets in case of cilia and flagella. So over here you will see that they are made up of triplets. So microtubules is the constituent again. Microtubule is the constituent again, but this time microtubule is the constituent again, but this time, you know what happens is they are present in triplets. Now, internal structure, we're describing this structure. So they are made up of, they have got this 9+0 arrangement. So why 9+0? Why the central part is absent? You can see the central part is absent, all right? So there are no central microtubules. This is not a microtubule. This is called the hub. I'm going to tell you about that. So there is 9+0 because they do not show movement. All right. So they do not show movement. So what is the use of that central wheel kind of a thing? So they just support the cilia and flagella, and centrioles, they split, and before cell division they split, and during cell division at the time of prophase they split and they give rise to the spindle fibers. So 9+0 arrangement means there are these 9 peripheral microtubules. No central microtubules. And the other thing is that they are present in triplets. So they are these 3 subunits made up of named as A, B, and a C. So they are made up of A, B, and a C subunit. So they are triplets. While A is made up of 13 protofilaments. I told you the filaments present in the walls of these units. So while there are 13 protofilaments present in the wall of A, and the B has 10, and C also has 10. So you may be asked in an MCQ about the number of protofilaments. And these protofilaments are made up of alpha and beta tubulin proteins. So microtubules are basically made up of tubule proteins. So the protofilaments, they are made up of the alpha and beta. I told you the structure initially while you were discussing the protofilaments of cilia and flagella. So over here also they are made up of protofilaments, and the protofilaments are made up of tubulin protein. But over here now, see over there that was 13 and 10. Additionally, there is C, and C is also made up of 10 10 protofilaments. All right. So first of all, they are present in triplets. Now, how are these microtubules arranged? How are these microtubules arranged? You can see this diagram over here. So you can see that there is this A, B, and C, and like A is position of A is interior. Then after that you have B, and then you have C. So there are no raised structures in A. And what kind of connection is there between the peripheral microtubules? A is connected to A of one triplet is connected to C of another. So you can see A and C are connected all the time. A and C are connected. You can see there is this A and C linker, AC linker. So over here you will have the AC linker. Can you just see AC linker? So A of one subunit. If this is A, it is going to be connected to. If this is A, this is going to be connected to C. So again, A to C. So if this is A, this is C, this is A. So wherever, which part you'll make A, wherever you have these spokes, you know. So this is A all the time, and you have C, which is connected to that. So this is C. Again, this is C. So they may ask you to label or something like that. So which parts you will label as A? You will label those parts as A, which have those radial spokes coming out. All right. So radial spokes will come out from A, and you can see these small structures which are present. You can see these small structures that are present. So first of all, triplet is clear to you, number of protofilaments clear to you, protein present in the protofilament is clear to you. Now, let us now see. Microtubule triplets are also made up of tubulin proteins over here, but over there that was dynein. In cilia and flagella, there was dynein and nexin on the linkers. This central part, this central structure is not made up of microtubule. It is called hub. It is called the central hub. And all the time you will see that there are these structures, these this triangle shape or structures. They are called X bodies. And from the X bodies, now there's this X body near the A subunit, and from the X body you have these radial spokes that originate. So here is the radial spoke. So this is the radial spoke. Okay. So and look at the diameter is of the linkers is the dimension is 10 nanometers. The dimension is 10 nanometers. So once again, let us just revise. But additionally, what they have, what is missing over here is there is this X bodies there. In between the X bodies, you have Y bodies also, and they are just linked. It just forms a cart kind of a structure. So A and B are linked. And see, there are two linkages. There is one linkage between, this Y is linked to, Y body is linked to the linker and also to the neighboring X's. So it's linked to the linker and neighboring X's. It's linked to the linker and neighboring X's. Again, this is linked to the linker and neighboring. So if this is the X body, this is the Y body. This is linked to the linker and the neighboring X bodies. You understanding? So what kind of linkages? Once again, let us do. Once again, we are revising the linkages over here. So you have 3 subunits A, B, and C. The first linkage is between A and C. A of one A subunit is linked to another C subunit by the AC linker. Then you have the radial spoke coming out from the A subunit, but in between the A and the radial spoke, there is this X body. And between two X bodies, there is this Y body. So X bodies and Y bodies are linked, and Y body is also linked to the AC linker. So Y body, if this is the Y body, this has linkage with the AC linker and also with the A subunits, all right, or the X bodies on the lying close to the A subunit. And this is the central hub. So it doesn't rotate like a wheel. Initially in cilia and flagella, that used to behave like a wheel kind, cartwheel kind of an arrangement. But here it does not rotate, all right. And you have the radial spokes. So this gives a supporting structure. So this is the structure of a centriole. This is the structure of the basal body. So these are the just supporting structures. They do not rotate. They just support. So this is the internal structure of centriole or basal body. All right. So is this clear to you?
Ma'am, the X body is also called the pinhead, right?
Yes, correct. So X body is called the pinhead, and in between the X bodies, you've got this Y body. So you understand the linkage. They ask you about the AC linkers, and they ask you about the AB linkers all the time to compare. So all the time, this is made up of tubulin protein, and then there is the central part does not have the microtubules. It has the hub. All right. So this is the structure of centriole, cilia, and flagella. So cilia, flagella, same internal structure, and basal body and centriole, same internal structure. Is this clear, crystal clear to you all?
Yes, ma'am.
Just look at it for a minute, for some minutes. So if you get a question, so what kind of questions did you encounter? You must have encountered questions about linkers, and you must have encountered questions about the radial spokes and all these. So what kind of an arrangement you will find in cilia and flagella? So if they ask you 9+0 arrangement is present in, so 9+0 arrangement, I'm giving you a question now. Listen carefully. 9+0 arrangement of microtubules is present in, you have to tell me, stereocilia, basal body, centriole, and the last one is all the above. What will you say?
All the above.
Yes, because stereocilia is non-motile, so it has 9+0 arrangement, and centriole has 9+0, whereas basal body also has 9+0. So this will be the answer. So all the time now clear, you should just mark all these structures, and so that before the NEET exam, just have a look and go through it. All right. So we are done with the structure. Now let us just quickly see the PPT on cell and see wherever doubts we have. Now there is one more very important thing that is called the GERL system. Have you ever encountered that GERL system?
Yes, ma'am.
What does that GERL system stand for?
Ma'am,
Golgi apparatus,
Golgi
endoplasmic
endoplasmic
reticulum,
reticulum, ribosome,
Okay, so
lysosome.
let us just look at the PPT and you can recall from there. So GERL system is something which is important, how the endoplasmic reticulum, Golgi body, and the lysosomes are related to each other. The lysosomes are related to each other.
Yes, ma'am.
Right. Yes, see, I wanted to show you this. Now, you can see cytoskeleton also is present in a cell, which is network of filamentous proteinaceous structures. So you have this cytoskeleton, and then microtubules, which is made up of contractile protein tubulin. We just discussed this. And as in case of centrioles, the microtubule, they form these spindle fibers. And What is microfilament? It is made up of that contractile protein actin. So microfilament is present in the muscle fibers. Microfilament is present in that muscle fibers, whereas microtubules are present in the centrioles and in the cilia and flagella. Now cilia and flagella, see, they have shown this. Can you see now? Whiplash kind, when the lateral hair-like structures are absent. So you can see whiplash. Whiplash may the diameter is not uniform and the hair-like structures are absent. I told you, that the bristle-like structures are absent in whiplash, whereas in tinsel type, they bear lateral hair-like structures called flimmers. And then there's this one cilia magnified. Now look at this. Two centrioles are at right angles to each other. So you can see the name, who discovered these centrosome and the centrioles. So Theodore Boveri had discovered these centrioles. They are also called diplosomes because they are 2 in number and at right angles to each other. So they are 2 in number and they are at right angles to each other. And you can see that the cytoplasm surrounds the. Now, we drew the diagram that there are two. You can see that there are two centrioles. They are right angles to each other, and the area of the cytoplasmic part around the centriole is called the centrosphere. All right. And each centriole is surrounded by, yes, and there is this pericentriolar mass also. You will find these dotted structures. There are approximately 9 in number. That is called the pericentriolar mass. It is also called masseules or crown or satellite. This is important. We tend to neglect these things. And now finally what we have discussed today is centriole mainly consists of 9 peripheral fibers of tubulin protein, and they show 9 plus 0 arrangement. So this is a very important NEET question. They also termed as cell centers. All right. So this is about your centrioles. Now I wanted to show you structure of chromosome also. Are you aware about nucleus's structure? Look at the structure of the nucleus. You can see this is an electron micrographic structure of the nucleus, where you can see that 2 distinct areas are there in a nucleus. There is this lightly stained euchromatin where most of the genes are concentrated. That's why it is genetically more active. So more genes are present over here, more genes. So they are active. That means more proteins are formed from this area. Heterochromatin is darkly stained. It is darkly stained, genetically less active because it has less DNA. So you do get to differentiate between heterochromatin and euchromatin. So heterochromatin is darkly stained, euchromatin is lightly stained. This is genetically active. This is genetically less active because less genes are present. All right. So heterochromatin and euchromatin, this is asked in the NEET exams. So NEET question this is. Be very careful while you're doing this. Now next is, yes, you know about nucleolus. Nucleolus helps in RNA synthesis, all different kinds of RNAs. What are the 3 different kinds of RNAs? 3 different kinds of RNAs are ribosomal RNA, messenger RNAs formed during transcription, you know that, and tRNA or transfer RNA. So all these RNAs except messenger RNA is made from the nucleolus. Messenger RNA is formed when? So inside the nucleus only messenger RNA is formed at the time of transcription, and all these 3 kinds of RNAs, after being formed in the nucleus, They move to the cytoplasm for protein synthesis. They move to the cytoplasm for protein synthesis. Okay. Next is, now nucleolus regions are not so important, and these are the constituents what the nucleolus is made up of: proteins, RNA, and DNA. Nucleolus, it helps in the formation of different kinds of ribosomes. That is the most important function of the nucleolus. All right. Now chromosome structure is very important. You do get questions from chromosomes because you do have genetics in your syllabus. So these terms, scientists who first described chromosome, means the answer will be Strasburger, and the term chromosome was coined by Waldeyer. Chromosomes literally means a stained body, and these chromosomes are basically, they are formed as a result of shortening and condensation of the chromatin material, and they can be best studied during metaphase. And look at the number of chromosomes. Okay, they do ask you this question. This is a NEET question. Maximum number of chromosomes. Ophioglossum reticulatum. Ophioglossum reticulatum is a pteridophyte. That means fern category it is. So Ophioglossum reticulatum has the maximum number of chromosomes. So the diploid number is 12,260, and the haploid number is 630, the half of that. All right. And you can see Muker's number. So Muker is just diploid. I mean, there's 2 chromosomes. They also ask you the number of chromosomes in Drosophila and also in P. This is Mendel's experimental material, garden P. So the number of chromosomes is 14 and the haploid number is 7. All right, you just remember maximum and minimum over here is the Muker fungi. And yes, particular species also Hemelis they have given. All right. Amongst animals, Ascaris has the lowest number. Ascaris has the lowest number. Drosophila melanogaster. This was used in linkage experiments by Thomas Hunt Morgan. By T.H. Morgan. So this has 8 chromosomes. So the diploid number is 8 and the haploid number is 4. Homo sapiens, everybody knows. And this protozoan Olocantha has 1600 chromosomes. This is also a NEET question. That you will mark. Okay. Now, based on the types of chromosomes, based on the position of the centromere. So when the centromere is in the center, this is metacentric. Metacentric when the centromere is at the mid. All right, submetacentric, it is like, little away from the center. So one arm is little longer than the other arm. So this is submetacentric. Submetacentric is going to be somewhat, L-shaped. It is going to be L-shaped. Then you have acrocentric, which is J-shape, where, acrocentric means it is not at the end. It is like, one arm is very long and the other is very short. It's like this. So this is the J-shaped kind of a chromosome, and this is acrocentric. So near the end is this one. Anterocentric is just at the one end. So these are the different types of chromosomes based on position of the centromere. So look at this arm ratio. The ratio of the length of long arm to the short arm gives you the arm ratio, and it is maximum in case of acrocentric, because in case of acrocentric, one arm is very long. So length of the long arms length will be longer, and the short arms length will be the least. So over here the ratio will be maximum. So this is also a very important question. You should just do this. All right. Again, you know the diagrams that they have given you. So telocentric, metacentric in the center, submetacentric little away from the center, so they will differ in their lengths a little bit. Acrocentric, one very long, one very short. This is acrocentric and telocentric. And karyotyping is a very important term, and that describes, you know, the number of chromosomes, the size, position, and centromere and length. You know what is secondary constriction? You should understand these terms: secondary constriction, primary constriction. The centromere is called, the other name of the centromere is primary constriction. So like if this is a chromosome and here is a primary constriction, another constriction apart from the primary constriction is called the secondary constriction, and it is also called the nucleolar organizer because it gives rise to the nucleolar organizer. Understanding? Karyotyping means when you are just you are placing all the chromosomes on the base, studying all the chromosomes on the basis of their size, position of centromere, length of arms, and the banding, the secondary constrictions and everything. And the diagrammatic representation is the ideogram. So karyotyping is just arranging them. But the diagram, when you see the diagrams of all the chromosomes on the basis of their size and position of centromere, etc., it's called an ideogram. Kinetochore is that point where the spindle fibers attach. So you can see this will be bead-like structures. So the structure of the chromosome, the two arms are called chromatids, and this is important over here. And each chromatid has DNA with histone protein. Histone proteins, because of this, DNA is positively charged. And chromomeres, they are bead-like structures. So if you have the, you know, chromosome like this, so bead-like structures on the chromosomes. These are chromomeres. So if you can just imagine this as chromosome before the interphase, you will have bead-like structures. These are chromomeres. Sister chromatids are connected. Now after the S phase, the chromosome duplicates. The point of attachment of the chromatids is called centromere or primary constriction. And at the centromere, there are two protein discs which are called kinetochore, and kinetochore is the point of attachment of the spindle fibers. So they are the actual sites. So this is a very important question for cell division. The actual site of attachment of spindles. Why centromere is called the primary constriction? Why centromere is called the primary constriction? The answer will be at the region of the centromere, the chromosome is comparatively narrower. So at this region, the chromosome is narrower and than the remaining part of the chromosome. So it is called the primary constriction. See, secondary constriction, I was telling you, it is also called the nucleolar organizer region because it gives rise to the nucleolus. Telomere is the end. Satellite is a part beyond the secondary constriction. Satellite is a part beyond the secondary constriction. Satellite is a part beyond the secondary constriction. Telomere is rich in guanine bases. This is also very important. So look at the structure. So this is this ground substance of the chromosome is called the matrix, and the outermost layer is the pellicle. So ground substance is the matrix, the outer layer is the pellicle. The beaded structures are called chromomeres. Initial constriction, where it becomes narrow, where the point of attachment is there and where you have these kinetochores, is the primary constriction. Additional constriction apart from the primary constriction is called the secondary constriction. This is the place. This is also called the nucleolar organizer region. And what is a satellite? The part beyond the secondary constriction is the satellite. I will tell you the importance of the satellite. End is called the telomere. End is called the telomere. Sometimes you have another secondary constriction too near the telomere. So is this structure clear to you? Is this structure clear to you? Just write on the chat box and send me.
Yes,
All
ma'am.
these are important questions. Is it clear to all three of you?
Yes, ma'am.
So these are all important questions. You must go through them. All right. Now you have this. Now next class, I'm going to explain you from here. I'm going to explain you this the nucleosome model of the the nucleus, the nucleosome model of the chromosome. I will explain you. DNA is so long. DNA is so long. How come it fits into the nucleus? So it's highly coiled. And what are the important components? You know, DNA has this the nucleus. Actually, they are organized into nucleosomes. So where they have this, histone proteins. There are histone proteins and DNA binds to them. So how come this association is possible, and what are the different kinds of histone proteins? As you can see, H2A, H2B, H3, H4, and they form an octamer. There are 8 subunits. So roughly you can just see, we'll start from here next class. So histone forms an octamer, and on that you can see, you've seen a wool ball, how the wool is, coiled and coiled on that ball. So similarly, you have this histone forms an octamer. It's a collection of 8 proteins. You cannot see this is that 3D view. This is not there. So this is this histone octamer. Histone, the 8 subunits collect, and on that DNA is wound. So this is DNA. They form the core. So next class we will start from here. We will discuss the structure of the chromosome. All right. So we will stop over here, and next class we are going to start from here. You just revise till here. So today, what all we have done the structure of cilia, flagella, centrioles, then basal granules. So cilia and flagella have the same internal structure. Basal granules and basal bodies and centrioles have the same structure. Then we did some facts about chromosomes, their numbers, their structure, species having or animals having the largest number of chromosomes, plants having or pteridophyte having the largest number of chromosomes, etc. All right. So next class we will continue from here, revise everything from NCERT. All right. And thank you so much for joining.
Thank you, ma'am.